If you take a quick look at this surface of the cerebral hemisphere and this surface of the cerebral hemisphere, what is the difference which immediately comes to our attention? We notice that this surface, we can see the sulci and the gyri much more clearly, and this surface seems to be covered by something. So what is the difference? The difference is that this side, the piamatter and its covering and the blood vessels embedded in the piamatter have been completely removed, and therefore we can see the surface of the brain very clearly, and on this side it is still to be removed. So what I planned is, while I'm going about the job of removing the piamatter on this surface, I'll give a running commentary on the piamatter, its various derivatives, and a few other topics which are not very frequently talked about pertaining to the piamatter. Now if you look very closely just on the parasagittal region on this side, we can see some small granular structures here and here. These granular structures that we see here, these are known as arachnoid villi, arachnoid granulations, and especially the large ones which are not seen here, they are called the Pacchionian bodies. What are these? These are actually derivatives of the arachnoid, and they are responsible for absorption of CSF into the superior sagittal sinus. Having said that, let's start going about the business of removing the piamatter. By the way, what is the meaning of the word piamatter? What is the origin of the word piamatter? The piamatter literally means soft mother. Pia means soft, mater is mother. Because as you can see, it is a delicate, thin, translucent covering of the membrane. It is nurturing to the brain. That's why it is called the soft mother. In contrast, the other two layers of the meninges, the dura mater and the arachnoid, are respectively called the hard mother and the spiral-like mother, but we shall not talk about those. So that's about the origin of the name piamatter. If you take a look at the gross appearance of the piamatter, you find that it is delicate, thin, and translucent. This is the only layer of the meninges which closely invests the surface of the brain. It dips into the sulci and comes out over the gyri. This has got a layer of blood vessels embedded within it. Therefore, it is nutritive to the brain. That's why also another reason why it is considered as the nutritive layer for the brain. Now, as I'm going about removing the piamatter from the surface of the brain, we should be careful about noticing one thing. This is actually a post-mortem artifact, a preservation artifact, whereby the piamatter is easily coming out from the surface of the brain. But remember, in actual situation, in a living brain, this is not possible. If we try to remove the piamatter from the surface of the brain, we will produce hemorrhagic spots on the surface of the brain, and the brain, that surface areas, will undergo gliosis. So how is it that we are able to remove it here? We are able to remove it here because an artificial space has been created under the piamatter, the so-called subpial space. As I mentioned, it is a preservation and a post-mortem artifact. So that brings me to this concept of subpial space. The subpial space is a microscopic space under the piamatter. It separates the piamatter from a layer which is present on the surface of the brain, which we cannot see grossly, but it is visible microscopically, and that is called the outer glial limiting membrane, or the glial limitans. This outer glial limiting membrane, or the glial limitans, is composed of astrocyte foot processes with an overlying layer of basal lamina. This outer glial limiting membrane is the one which is separated from the piamatter by the subpial space. So what happens in a normal brain, in a living brain, if we try to remove the piamatter? What will happen is, we will produce hemorrhagic spots and contusions on the surface of the brain, and that is exactly what is the pathophysiology of contusions. But in this case, as I said, because an separation artifact, post-mortem artifact has been created, that's why we can remove the piamatter relatively easily. Having said that, I would like to draw your attention to something else. As I'm removing the piamatter, we can see that it contains blood vessels embedded inside it, and we can see the blood vessels here. What happens is, these blood vessels, they are embedded in the piamatter, and as the piamatter dips into the sulci and the substance of the brain, the blood vessels also penetrate in. Surrounding these blood vessels, there is a small microscopic space which is referred to as the Furcault-Robin space, which is present only in certain areas of the brain. And these Furcault-Robin space again cannot be seen grossly, but they are visible. If they are large, they can be visible in MRI. These Furcault-Robin spaces, this is a blood vessel by the way, these Furcault-Robin spaces have got many, is the topic of very hot research, and it has been postulated that this Fourcault-Robin space may be one of the ways of removing the extracellular fluid from the surface of the brain. Okay. There's another concept which I want to highlight when I'm talking about the piamatter. This piamatter, which was there on the surface of the brain, most of it has been removed here. This piamatter is covered by a layer of arachnoid, and between the two there is CSF. So this piamatter provides what is known as the outer CSF brain interface, meaning thereby that CSF can circulate through the piamatter. The piamatter is composed of flat mesothelial cells with spaces in between the cells. So the CSF can percolate through that into the subpial space. From there it can go through the basal lamina, which I mentioned earlier, and it can go between the astrocyte foot processes, and it can reach the brain. Therefore, that is known as the outer CSF brain interface. Microscopically, this piamatter is composed of just a layer of flat mesothelial cells, as I mentioned earlier. This is a look at some of the blood vessels as they are penetrating into the substance of the brain, and they are all enclosed in a piamatter. So in the process of removing the piamatter, what we are doing is we are essentially removing the blood vessels also. Let's take a look at contusion. How in real life, what exactly happens when there is a contusion? Suppose the brain is jolted inside the cranial cavity very hard. There is no tear of the dura or arachnoid, but what happens is the piamatter gets rubbed off from the surface of the brain. And once that gets rubbed off, hemorrhagic spots appear on the surface of the brain, and these hemorrhagic spots then get converted to hemosiderin, and later on they can form astrogliosis, and they can be focus of epilepsy. This hemosiderin at a later stage will become yellowish brown plaques, which are known as the plaque jaune. So this is a few things about the piamatter. Last but not the least, there are a few derivatives of the piamatter which I would like to talk about. What are the derivatives of the piamatter? One derivative of the piamatter, which is not seen in this particular specimen, but I will show it to you in this hemisection of the brain. If we take a close look at this structure here, this structure, this is what is known as the choroid plexus of the third ventricle. This choroid plexus of the third ventricle is actually formed by a leash of blood vessels embedded within a double layer of piamatter, and this double layer of piamatter is known as the tela choroidea. So this choroid plexus, which is composed of the tela choroidea and the ependyma, is the secretory apparatus of CSF. So this is one derivative of the piamatter. Another derivative of the piamatter, which again is not visible in this or this specimen, that is called the denticulate ligament, which is present only in the spinal cord. There are 21 pairs of projections from the spinal piamatter which tether the spinal cord to the dura inside the vertebral canal. So these are some of the derivatives of the piamatter. And finally, the last point, what are the functions of the piamatter? Function of the piamatter is nutritive to the brain, supporting the brain, providing blood vessels to supply the brain, and it is also, in a certain measure, responsible for maintaining the metabolic environment of the CSF and the extracellular space of the brain. So with these few words, we shall conclude our discussion about the piamatter, and now you can see that we have removed the piamatter from both the surfaces, and we can see the sulci and gyri very clearly. This is an essential first step that we have to do before we study the surface of the brain. We need to remove the piamatter so that we can see the sulci and gyri clearly. Thank you for watching. If you have any comments or questions, you can put it in the comment section below.